We've now discussed both the structure of DNA as well as the chromosomal structure. And now we're going to talk about DNA replication, the passing on of your genes and chromosomes to daughter cells. As we begin this topic, the first question we should ask ourselves is, why do we even need DNA replication? And for a second, I'd like you to think about situations in your body when you might need to replace a damaged cell. So if you're to think about possibly wounding your arm, let's say you were riding your bike, you fell off your bike, you scraped your arm, you're going to develop a scab, and hopefully the skin that had been there, which is now scraped off and left on the street, will be replaced by new skin cells. So as we're talking about DNA replication, you can continue to think back to this example. So what we need to do is to create a new skin cell to replace the skin cell that has been sloughed off or lost in our accident. So again, we're going to look at the structure of DNA, since this is what we want to replicate. And I'd like for you to think about some of the key features of DNA. We know that it's double-stranded. We also know that it has this helical structure. We know that there are 4 different nitrogenous bases that are linked together by hydrogen bonding. Again, the adenines bind with the thymines with 2 hydrogen bonds, and the guanine are bound to the cytosines by 3 hydrogen bonds. And it's this hydrogen bonding pattern that is keeping the 2 strands of DNA linked together. We know that hydrogen bonds, as we discussed in chapter 1, is a relatively weak bond and quite easily to break apart. So again, here's showing you the binding between adenine and thymine, which is 2 hydrogen bonds, and the bonding pattern between cytosine and guanine, which is 3 hydrogen bonds. So as we're beginning to discuss DNA replication, we should begin with an overview of this overall process. So we begin with our original double helix that we want to replicate. Somehow we need those 2 strands to separate. We then need to bind complementary base pairs to those strands. And then we're going to need to link up the different nucleotides together to form a cohesive helix. As we begin to synthesize a new strand of DNA that's complementary to the parental strand, we need to recognize that this whole process can only take place due to a particular protein or enzyme called DNA polymerase. And DNA polymerase is able to help polymerize or synthesize that new strand of DNA. So as shown here in the picture, when we have opened up that double strand of DNA and now we want to form a complementary base pairing with the parental strand, with one of the parental strands, the complementary base pair is going to link up to the parental strand. So if you look at the DNA on the right-hand side of this picture, you can see that a cytosine has formed a triple bond with the guanine. However, that cytosine is not linked to the backbone of the DNA structure that already has a cytosine and an adenine bound there. So in order for us to form that sugar phosphate backbone, that process is facilitated by DNA polymerase. And one of the key ideas here is that DNA polymerase can only polymerize DNA in the 5' to 3' direction. If you look at the DNA strands, you can see that there actually is some polarity to the strands. And we, by convention, call the carbon that's attached to that phosphate at the top the 5' end. And we call the hydroxyl group on the bottom of the sugar of the deoxyribose, that's called our 3' hydroxyl. So we're adding DNA at that 3' end. So here is showing what this whole overall process would look like. In blue is depicting the initial parental strands that we have to pull apart in order to make a copy. And then the 2 daughter strands are shown here in red. So as I said, DNA polymerase can synthesize DNA easily in the 5' to 3' direction. But the problem is that when we've pulled apart these two strands of DNA, one strand of DNA is in the 5' to 3' direction, and the other is in the 3' to 5' direction. So when it comes time to synthesizing that new DNA, DNA polymerase can only work efficiently in one direction. So the way that we've been able to compensate for this is that on the lagging strand, the strand where DNA cannot polymerize in the 5' to 3' direction, what we do is polymerize DNA in short fragments that ultimately get linked up together. So when we want to synthesize new DNA, the first thing that needs to happen is we need to separate that initial parental strand, in this image shown here in black. So there is an important enzyme called helicase that is able to unwind the parental strands of DNA, basically breaking those hydrogen bonds. Now, if helicase just did that and then let go, what would happen is that the strands of DNA would go back and recoil. So what is necessary is single-stranded binding proteins, which are tiny proteins that come along and bind to the DNA and keep them separated so that new strands of DNA can be synthesized. So on the leading strand of DNA, DNA polymerase is now able to come along, is able to synthesize DNA in the 5' to 3' direction and does not have any issues. However, on the lagging strand, the DNA synthesis is much more complicated. The first thing that happens is primase will come and lay down a short primer of RNA. So it's just a few nucleotides, but it's actually RNA as opposed to DNA. If you recall, RNA, it has a ribose sugar as opposed to a deoxyribose sugar. But it has the same type of complementary base pairing that we see with DNA. DNA polymerase 3, which is the polymerase that synthesizes DNA in the 5' to 3' direction, is then able to come and polymerize a short fragment of DNA. At that point, once it hits the preceding RNA primer, the DNA polymerase will dissociate and fall off of the DNA. But we now need to replace that RNA primer, which is done with a different DNA polymerase called DNA polymerase 1. So DNA polymerase 1 will come and excise that RNA primer and will lay down DNA. The final step is there's one gap between these different fragments of DNA that have been synthesized. These fragments are called Okazaki fragments. And this final step is a linking of all the fragments, these tiny fragments, and that is done by DNA ligase. So again, what I'd like for you to do is pause the slide right here and see if you can define these key proteins involved in the synthesis of DNA on the lagging strand. So hopefully you were able to determine that DNA helicase is responsible for breaking up the hydrogen bondings between the DNA parental strands. Those single-stranded binding proteins are then able to keep the parental strands apart. Primase synthesizes a short RNA primer. DNA polymerase 3 then synthesizes a short daughter strand of DNA. DNA polymerase 1 can then come and get rid of the RNA primer and fills that in with DNA. And DNA ligase seals up the Okazaki fragments, binding them to one another. So one question to think about is, how does the DNA know where to begin DNA replication or DNA synthesis? Can this replication occur anywhere throughout the chromosomes? So it depends upon what you're talking about. Are we talking about prokaryotes or eukaryotes? For prokaryotes, which is bacterial DNA, DNA replication is very controlled and only begins at a single origin of replication. So as you can see here, it begins at the origin of replication and it spreads out from there, and slowly that replication bubble gets bigger and bigger and bigger until at the end of DNA replication you have two circular pieces of DNA that are detached from one another. For eukaryotic DNA, this is not the case. We have much more DNA and we also have chromosomes, so it becomes somewhat complicated. So we have multiple origins of replication, and what you can see is we can have multiple replication bubbles where DNA is occurring simultaneously, such that eventually these replication bubbles will bump into one another until we have a completely duplicated chromosome. So here is a thought question for you. My question to you is, do you think this chromosome that's undergoing DNA replication, do you think this is a prokaryotic cell or a eukaryotic cell? Go ahead and pause the video and answer this question on a sheet of paper. Take a minute to think it through. Okay, hopefully your conclusion was that this is indeed a eukaryotic cell, because we have multiple origins of replication as opposed to just one origin of replication. You can see that we have several replication bubbles that are connected to one another. So the next question is, which site started undergoing DNA replication first? Site A, site B, or site C? Take a second to answer that question. Hopefully you concluded that site C is where DNA replication began. This is the largest of the three bubbles, hence DNA replication has been going on the longest at that particular site. So in conclusion, for this part of the chapter, we were able to learn that DNA replication on the leading strand involves DNA polymerase III and is quite simplistic as compared to the lagging strand, which involves a primase laying down the RNA primer, DNA polymerase III, DNA polymerase I, as well as ligase. For prokaryotes or bacterial cells, they have a single origin of replication, whereas for eukaryotes, we have multiple origins of replication. This ends this portion of Chapter 6.